Hello guys, welcome to the YouTube channel of Gilbert Professional Classes. Hope you all are doing good. So in today's tutorial we are going to start a different type of point mutation, that is the missense type of point mutation. So today we are going to start missense type of point mutation, that is commonly known as missense mutation. If we see the definition of missense mutation, you can write it like this: any change in a nucleotide pair that may transform one codon into another, that is translated into a different amino acid. That means missense mutation is a type of point mutation. Here, due to point mutation, one nucleotide pair will be changed from the gene level, and because of the change in that single nucleotide pair, the entire codon will be changed in the mRNA. And suppose before mutation the codon in the mRNA was XYZ, and this was the codon in the mRNA, suppose before mutation, and it was coding for an amino acid, suppose amino acid 1. Now due to mutation, suppose here the Y is changed into Z, so the codon now becomes XZZ due to mutation. After mutation, suppose here missense mutation occur. Now due to this change in codon, now the new codon formed after mutation is suppose coding for a different amino acid, suppose amino acid 2. So this type of mutation where the codon is changed due to mutation, and due to change in codon the amino acid is also changed, that is known as the missense type of mutation. Here codon is changed, and due to change in codon the amino acid is also changed. So that means here protein will be changed. So this type of mutation is known as missense mutation. So what we can conclude here the codon is changed due to mutation, and along with the change in codon the amino acid is also changed. The amino acid is also changed due to the mutation process. So if the amino acid is changed, that means we can say the missense mutation has the ability to affect the functioning of a protein. So it can affect the protein, because whenever the amino acid of a protein is changed, the protein properties will be changed. So that's why the protein function will also be changed. So missense mutation may change the functioning or the functions of a protein. Now we can understand the process by taking an example. So let's take an example. Suppose we take an example. Suppose in this side, on the left side, we are considering a wild type condition. Suppose we have considered a small DNA strand, double stranded DNA. Suppose this upper strand runs from 5 prime 3 prime direction. It is T A C T T, then C A C C Z A T T, and this is the 5 prime. So its complementary strand will start from 5 prime direction. It will be A T, then Z A, it will be Z, then T T T, then G Z C, Z Z C, then it will be T A, and this is the 3 prime. So this is suppose a double stranded DNA. Now in this double stranded DNA, suppose this double stranded DNA undergo the process of transcription. So it undergo the process of transcription, that means it is going to form the mRNA. Suppose in this process the upper strand is considered as a template. If the upper strand is considered as a template, then the mRNA will be like this: 5 prime, it will be A U Z, then A Z, then U then G G C U A 3 prime. So this is the mRNA if the upper strand is used as a template. Now in this mRNA we are getting different codons. Suppose this 3 represent one codon, then this 3 represent another codon, next 3 represent another codon, these 3 represent one codon, and these 3 represent the codon. Now if you consider the last codon here towards the 3' end, this last codon, it is actually a stop codon, so it will not code for any amino acid. However, the remaining codons, they will code for amino acids, and those amino acids form peptide bond in between them, and they are forming one particular protein. Now if this mRNA is translated, it is going to form a protein. So in that protein, different amino acids will be there. If you consider the first codon AUG, that will code for amino acid methionine. If you consider the second codon AAZ, that will code for the second amino acid, that is lysine. The third codon UUU, that will code for the amino acid phenylalanine. And if you consider the fourth codon, that is ZZC, that will code for the amino acid glycine. But the fifth amino acid, that is a fifth codon, that is a stop codon, that will not give any amino acid in this case. So suppose this is the N terminus of the protein and this is the C terminus. Suppose in this wild type case, the protein is there where the amino acid, 4 amino acids are present in the sequence: methionine, lysine, phenylalanine, and glycine. Okay, this is suppose a protein after translation in the wild type condition. Suppose in this wild type DNA, one point mutation occurs, and due to that point mutation, suppose the point mutation actually occurs in this particular position. Suppose in this particular C, this particular C is suppose changed into T. Okay, so in that case what will happen? Suppose we are drawing the mutant type here. The mutant type will look like this. Mutant type here, the 3' end, then you will get TA, then CTT, you will get C triple A, then you will get here T. So as it is changed, this is the point mutation, I am changing the color here, it is T instead of C. Okay, then you will get again C, then G, A, then T, T, and this is the 5' end. This is the 5' end. Okay, now its complementary strand will be 5' A, T, then it will be Z, A, Z, then you will get T, 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 then here it is changed, that's why I am changing the color, it will be A, then again it is G, C, T, A, and this is the 3' end. So suppose this is the double stranded DNA after mutation. So here only one nucleotide pair is changed, and this nucleotide pair is changed, and it is now TA. It was earlier CG, and CG pair is changed into TA pair. Okay, now suppose this particular double stranded DNA undergo the process of transcription, that means it is giving rise to mRNA. So in that case, suppose the upper strand is acting as the template. If the upper strand is acting as the template, so the mRNA will be like this. It is 5' AUZ, then you will get AAZ, then you will get UUU, then you will get AZC, then you will get UAA. So here this particular A is actually the mutated version, so that's why I am changing the color here. So this is suppose the 3' end of this particular mRNA. This is the mRNA after transcription. Now in this mRNA we will get different type of codons. Okay, so this is one codon, these 3 forming the next codon, then the third codon, then this is the fourth codon, and this is the fifth codon. If you consider here fifth codon UAA, this is also a stop codon. Here also it is a stop codon, it will not code for any amino acid. If this mRNA go for translation, this will translate into a protein. It will go translate into a protein. So in that protein the amino acids will be present. The first codon will code for the first amino acid, that is methionine here in this case. Then the second codon will code for lysine. Then the third codon will code for phenylalanine. And here the fourth codon is changed due to mutation. Earlier it was ZZC, now it is AZC. So earlier it was coding before mutation ZZC, so it was coding for glycine. Now after mutation it is AGC, and AGC will not code for glycine. Instead of glycine it start coding for serine. So that's why I am changing the color here. It is now it is serine. So this is the N terminus, this is the C terminus. Now this is the protein after the translation of the mutated mRNA. So here what we have seen that in case of wild type case, the protein was made up of 4 amino acids in the sequence methionine, lysine, phenylalanine and glycine. But after mutation, the protein that is changed and it is now methionine, lysine, phenylalanine, and in place of glycine, it is now serine this time. So as the amino acid is changed for this protein after mutation, this protein functions will also be changed. This protein is no longer the same protein as the mutated protein. Okay, its functions also changed, its properties are also changed. So this type of mutation where the change in codon, this codon and this codon, here change in codon leads to change in amino acid. Here this amino acid is changed and after mutation this amino acid is changed. So this is known as the missense type of mutation. So there is one practical example of this missense type of mutation, that is the beta globin mutation, as we have seen in case of the as we have seen this type of mutation in case of our sickle cell disease. In the sickle cell disease, actually the beta globin is mutated, and due to that mutated beta globin, the entire hemoglobin, adult hemoglobin, is changed into the sickle cell hemoglobin, that is HbS. And due to that, ultimately RBC shape is changed into sickle shaped, and that's why it is developing the disease sickle cell anemia. So that is one kind of practical example of a missense mutation, and that example is already discussed in our previous videos. If you are new to our channel, you please go back and see our previous videos, you will get the concept. Okay, so that's all from the missense type of mutation. Hope you have understood the video. If you like the video, please subscribe the channel and share it with your friends. Thank you.